adventuring with the other fathers and sons that came. This week, some of you have noticed there's a big chart over there, and unfortunately we don't have a table nearly long enough to stretch it all the way out. But if you want to take a peek at it afterwards, it was done by a man named Adams in the 1870s, I believe it was. But it's a chart of all of history reaching back to Adam. And you can kind of see connections there where the lines of people that descended from Adam, and then the flood, and then the spreading out of people groups, and we start to develop kingdoms, and then as we see those spread and become things that are familiar to us, things we've heard of like Babylon, the Roman Empire, Russia, I mean, they're all there, okay? And it just serves to show how all of history is documented, okay? So feel free to check that out afterwards if you haven't already. I think some people have been interested in it already. This is week 6 out of 8. This week we're talking primarily about genetics. And so we're going to dive into that in a minute. But if you remember 2 weeks ago when we were last together, we covered how modern science has left the evolutionary narrative without much footing, in spite of the continued attempts of popular media and public schools to suggest otherwise. So this week we're going to be discussing modern genetics, and we're going to briefly touch on how a basic understanding of genetics directly contradicts Darwin's proposed theory. Then we'll discuss how our emerging understanding of genetics points us to the vast creativity displayed in the adaptability of creatures. And then we'll see how the most recent developments in human genetics point us back to biblical history being factual. So this week, it is fairly science heavy. I'll say, if your eyes are starting to glaze over, what I really want you to be thinking about and getting out of this is, I want you to understand that there's immense complexity and creativity in the engineering of something like you and I. And then I want you also to understand that the evidence does suggest a biblical timeline. So watch for those themes here, okay? So it's all in the genes, right? I mean, this is a phrase I heard so many times growing up. And so many people have attempted to locate the gene for many kinds of things like personalities, illnesses, lifestyle choices, etc. But what we've learned now is that our genes are not deterministic. They only serve as information storage. In fact, we have many genes in us that are turned on and off at various times, some during growth and maturation, and others when we need them. And now we're moving kind of beyond this understanding, realizing that genes are not deterministic, but in fact storage of information, and we're looking for how and why they turn on and off and stuff, and we call this epigenetics. And we're really kind of in our infancy as far as understanding epigenetics, but epigenetics, if you hear that term, is the study of how genes are regulated. But the reason I bring that part up, I just want you to realize that once again, the deeper we look, the more complex our biology truly is. See, if you got here a little early, we listened to Dr. Paul Nelson, and he was discussing how much more information processing is in a single cell than in a supercomputer. In this clip, we're going to hear from Dr. Robert Carter, who's a marine biologist and geneticist, about how DNA is vastly more complex and superior coding to anything we can come up with in modern computers.
I like to say that the genome is 4-dimensional. In software, we write in lines of code.
Correct.
Well, in mathematics, we learned a line is a 1-dimensional object. It just has length. So you can actually take a computer program and just write from left to right for millions of characters. Now, on our put carriage returns in there so we can read it. But the computer doesn't know that. It ignores the carriage returns. It's just a line. Well, DNA is a line. So in the naive concept of DNA, we had a line that had information in it. But it's not simple like that. Because this piece of DNA makes a little protein that comes over here and sticks on this piece of DNA over here, which turns on or turns off a gene.
Oh my goodness, it's like self-modifying code.
Oh, it's worse than that. Because this piece of DNA over here makes just an RNA that goes over here and interferes with this gene's RNA. They stick together, they interfere, they conflict with one another, they turn things on, they turn things off. And if you want to draw that out, you need a sheet of paper, a very big sheet of paper. You'd have to write all the letters of DNA out on all 3 billion of them. It would take, I calculated, about 850 Bibles. Is one human genome. And then you have to draw lines or arrows from one part to another part, because this part turns this part off, this part interferes with this part enhances this. It's this huge two-dimensional interaction network. That's why you have a two-dimensional genome.
So it sounds like, let me stop you for a second, because this is really amazing to think about this, because I think in terms of a computer program that it's fairly static. I mean, instructions are there, but you're talking about a program that is reprogramming itself. It's modifying its own instructions.
Well, wait till I get to the 4th dimension.
Oh, okay.
Because of the 3rd dimension first. The genome also folds into a 3-dimensional shape. And so a 3D, the 3rd dimension, is actually the shape. And the genes that are buried inside this ball of DNA, they're not active, they're turned off. The genes that are exposed are the ones that are used. So whoever programmed that string knew when it folded up which genes would be available at what time.
I see. Are you saying that when this instruction set folds onto itself, it creates a whole new set of instructions?
Yeah, absolutely. And
That's my book.
the information in that 1st dimension, that linear string, has to be organized in such a way that when it folds into the 3rd dimension, it still works.
Oh, that's amazing.
But it's amazing. When they first sequenced the human genome, sometimes scientists sat down, they did something I would have done. They said, okay, let's look at genes that we know are used in a biochemical pathway. They might like 10 genes in a row to convert this into something over here. Well, if I was programmed it, I would have stuck them right next to each other in the genome. So they looked, and they weren't next to each other. They're random. They said, these genes are used on different chromosomes, they're backwards, they're forwards, they said, look at all the evidence of evolution, it's just junk. Random changes over millions of years, throw all this stuff together, willy-nilly, it's just nonsense.
And we've heard that a lot.
We've heard that a whole lot.
Junk DNA.
Yes, but then someone figured out how to look at the genome in 3 dimensions. First of all, they realized the genome folds in a fractal pattern. And it's beautiful. But it's in a fractal pattern that doesn't make knots. And so it folds up, and when they figured out where the genes were, genes that are used together are next to each other in 3D space. Even if they're on different chromosomes. When the chromosomes fold, they bring those 2 genes next to each other, and usually this cluster of genes is right next to a nuclear pore. So when God programmed these genes, he knew that when he had to turn all these genes on, he needed them in 3-dimensional space next to each other. So the whole biochemical pathway can be turned on. The little things are copied into RNA. The RNA comes outside the nucleus, is turned into protein. Voila.
That's amazing. Okay, so you're about blowing my mind with that, but you said there's another dimension.
Oh yeah, the 4th dimension is time.
And how does that work?
The genome changes shape over time. Remember I said that some genes are buried?
Yeah.
And some are exposed. Well, you need those buried genes at some time. And so at different stages of development, or sleeping versus waking, or stress versus non-stress, or after maybe you eat something that's bad for you, and your liver says, I can get rid of that toxin. Now, your earlobes, they don't care, they don't know what to do, but your liver says, I know what to do. The chromosomes in the liver will change shape, expose that new protein gene, make copies of it, build a brand new protein that can kill off that toxin, and when it's not needed anymore, they'll change shape again and fold back.
Right? Isn't that amazing? You know, I think this lends perspective to us on some of these passages. Like Isaiah 55, 8, 9. Who has that?
For my thoughts are not your thoughts, neither are your ways my ways, declares the Lord. For as the heavens are higher than the earth, so are my ways higher than your ways, and my thoughts than your thoughts.
Thanks. Yeah. You know, we've all heard that verse before. But man, it really means something different when you see a real example of how much higher the Lord's thoughts are than ours. Transcendent creator, indeed. All right, so let's talk about the genetics of adaptation. You see, there's no disagreement between evolutionists, and I say old Earth evolutionists, and young Earth creationists about this. Everyone agrees that creatures do adapt. The disagreement arises about the timing and the cause of adaptation. Old Earth evolutionists, of course, believe that mutation causes changes to organisms, and those changes, which are an advantage in the environment, lead to adaptation of a species, while the young Earth creationists might believe otherwise. In some ways, I would say our model of adaptation is just coming together in the last few years, but we're going to sort of reveal that here as we walk through the next few slides. But what does modern genetics demonstrate about how adaptation occurs? And which model of human history does this science fit? You see, mutation is an error. It's an error in the DNA, right? Here's a normal gene, a mutated gene. It's not quite right. Something's wrong here. And what happens with mutations, there's different kinds here we'll talk about in a minute, but most importantly what happens is, and if a mutation occurs in an area that codes for a protein, we get either an abnormal protein or no protein at all. You see, this can happen for a lot of reasons, but many mutations really can be harmless. They don't really seem to affect us much, but others cause problems, and sometimes many diseases that I learned about in medical school will say are based on mutations being passed on to children. Something like hemophilia, for example. But there's never been a true scientific example of a mutation that causes new information to be added to an organism. Now, we talked about this, but DNA is stored information. Okay, so let's use some information here. If you've seen this sentence before, it's great for testing fonts because it has every letter of the alphabet in it. But we're going to use this sentence as an example, and we're going to do a couple things here. Here we've done a quick substitution mutation, where we substituted a single letter out of the code. And you can see, you can kind of pretty much understand what's going on here, right? Not a lot of information is lost. You can kind of figure out what it was supposed to be. And this is a single nucleotide substitution mutation in the form of a sentence. So here we have something else. We have a single insertion mutation. And see, much like a sentence, the letters in DNA are in groups, kind of like words. And so if we do an insertion right here and add this I into the word, we now have a huge loss of information because the grouping of the letters doesn't change. And so you can see that this definitely causes a problem. Same thing here, this is a single deletion. We've taken the I out, and the grouping again doesn't change in DNA, so we lose all this information here. And so these kinds of mutations can be pretty harmful. So, anybody ever heard the word beneficial mutation thrown around? Maybe in school or something like that? I see some heads nodding yes. So, there's a couple things that people bring up and say, look, this is a beneficial mutation. One of them is sickle cell trait improves the resistance to malaria. But you should know that this is a loss of the normal gene for hemoglobin, and it causes devastating illness, severe pain attacks, organ failure, and even with modern medicine, the life expectancy of someone with sickle cell anemia is only around 50 years. I've seen sickle cell attacks myself and treated them in the emergency room. Let me tell you, this is not a beneficial mutation. Another one they bring up all the time, drug-resistant bacteria. And this is usually caused by a loss of a normal function that the bacteria has. So some enzyme gets poisoned, and now the antibiotics can't work via that enzyme anymore because this bacteria doesn't have it. And then sometimes there's another way that bacteria can actually share information, share DNA across from one species to another. And this is how drug resistance forms, but it's not. We're not generating new information that is somehow benefiting these bacteria. They're getting it from somewhere else or simply breaking something. And so you see mutations result in loss of information, loss of function, but never have we found one that adds information or adds function. So no, mutation does not create information. Does mutation help us create new forms? Well, let's listen to this interview. This is an educational administrator, and she's talking about classroom experiments that are designed to teach kids as evolution and mutations. She's Australian, so just be aware she's going to use the word prac, and she means a practical or hands-on lesson, okay?
I do remember trialing a prac once where we tried to introduce the idea of mutations, and mutations are actually random, they're not directed. You know, an organism can't choose to have a certain part of its genome deleted or extra information inserted.
Right, it's completely random.
Yeah, mutations are random. So we tried to do a prac to show randomness of mutations, and you'll have to try to visualize this with me. So we like birds apparently in science classrooms. So we did birds, and if we changed, mutations on where their wings are positioned, their wing length. So we did that by making these paper birds.
Okay.
So just straws and strips of paper.
Yep.
And we designed one that actually flew really well. And so that was our standard point. So we knew that it could fly. So then the prac was introduced to the students with dice. So, roll the die, and if you get, these numbers, you have to move the wing point on the piece of straw a centimetre to the left. And if you roll a number on this die, you have to reduce the size. So it was all just randomness in like what was going to be changed.
So accurately representing what mutations would kind of do.
Yes, exactly. And then so the criteria was that the bird had to fly a certain distance to say that it would be able to survive in the wild, because that's always sort of the clincher in terms of mutations and natural selection. Is something can change, but whether it's beneficial to that organism is a completely different matter. So unless it can survive to be able to reproduce, those traits won't be passed on. So we had sort of a distance this thing had to fly to say it could be passed on. What we found, but the prac failed spectacularly, because we soon discovered that with random changes, all the birds died, because we designed the bird obviously to be able to fly to start with. But as soon as we started changing that randomly, and by random amounts and random numbers, the data that came back at the end of the class was like, well, all the birds died, so if you changed anything, it didn't work. And so we quickly removed that from our repertoire of pracs and decided to delete that entirely, because it actually, it worked beautifully from a science point of view to show that mutations pretty much are detrimental to an organism. They're
So this experiment here, it seems like it's actually represented very accurately what happens.
Yes, but it doesn't help the evolution storyline.
Okay.
Yeah, if you notice there in her description, even those first paper birds that they were using, the ones that worked to start with, they had to be designed in order to be able to fly. And then, of course, all random mutations were detrimental. But see, this demonstration illustrates exactly what we are learning from genetics. In fact, in studying our own human genetics, we see that not only are mutations not creating new information or variants, but they're also not leading to improvements at all. Now, this is a Discover magazine article from 2013, and this is a study by evolutionists who are examining our genome, and they found that about 3/4 or so of mutations have occurred in the human genome, that the mutations that we have that are there have occurred within the past 5,000 years. And then they discovered that they think probably more than 80% of those are harmful to us. You see, so mutations not creating improvement. Are mutations then improving us? Well, let's listen to Dr. Don Batten explain.
You see, saying we as the human species are heading for genetic death.
We're heading for genetic decay is destroying us, not making us. And so time is the enemy of evolution. It doesn't save the problem.
So the answer is it's going the wrong way.
Going the wrong way.
Right.
Going the wrong way. Going backwards, not up. So, the Russian geneticist Alexei Kondrashov said, no, he's just an evolutionist, he said, why aren't we dead 100 times over? Looking at these mutations in humans, why aren't we dead 100 times over? Now, he's thinking about a time frame of 1 million years for humankind, and so he's got this concept in his head, so he's thinking about, with 1 million years, surely we should be extinct already. Because if we've only been here for 3,000 years, I mean, it makes sense that we're not extinct yet, but we're heading for extinction. So mutations are not creating us, they're destroying us.
See, our mutations are accumulating at a rate where they're actually killing us. So the science on this says that there's somewhere around 60 to 100 new mutations every generation. And that means from me to these two here, and their children will have even more. And these are accumulating at such a rate that in fact, there's no way the human species could survive for even 1 million years, as he pointed out. So, mutations are bad, right? So what about the process of natural selection? Could that work? Could natural selection actually evolve things? Well, let's first try to imagine what would happen if this were correct. And you'd see this is where that basic understanding of genetics helps. But this is a diagram of natural selection to teach you what natural selection does. And I like to start at the bottom and work my way backwards, right? So we have, generations here of organisms. And here at the bottom we have all either dark gray or black, right? Those are the only two kinds we have here. Now, if we back up here, we see what happens is back in this generation, we had 3 different kinds of organisms. We had those that are gray, those that are dark gray, and those that are black. And of course, you see that the dark gray ones make only dark gray ones, and the black ones make only black ones, and the gray ones make only gray ones. But we had all 3 at this point. And if we back up further, we see that originally, we had enough genetic information to make white and gray. And then this one here, this parent, if you will, had the information to make both dark gray and black offspring, right? And if we back up further, we go to the top of the chart, and we see that this parent here contained all of the information, genetically speaking, to make 4 different varieties. And so what's happened as we go down this chart is some of those varieties are lost, and we get to the bottom, and the only things that are living are 2 varieties that are left, and they only can make offspring that look like themselves. And so this is the actual process that natural selection would use if it was a real thing, because you are cutting off genetic information at every change, if you will. And so, genetically speaking, this is a process that causes a loss of variety. So not a gain in variety and new information. I mean, in fact, here, let's discuss, is natural selection even a valid idea? If you want, I'll put a link to this video at the end, and you can scan the code and watch this. But these four scientists discussed this idea for almost an hour, and we're going to listen to just 2 minutes for the sake of this class.
Yeah, and following on right onto that, we actually had a recent discussion just last year from another intelligent design advocate who was actually an agnostic, and the evidence kind of won him over. His name is Neil Thomas. He's a linguist at the University of Durham, or he's a retired professor now. He pointed out that Darwin, against the objections of Wallace and other colleagues who pointed out to him that there was simply no comparison between what animal breeders did by the use of human ingenuity and how mindless nature herself acted, claimed an analogy between the artificial breeding methods of such persons as pigeon fanciers and the claimed selection performed by nature herself.
And interestingly, it's worth noting the title of that article. It's Natural Selection, a conceptually incoherent term.
That was really what caught my attention here.
Yeah.
And just to fill in, the reason why, which we were not even going to discuss here, but that he was bringing up while it was conceptually incoherent, is Darwin's book, Origin of Species, was to be translated into German. So it had to go from one language to another. And you have to talk about nature selecting, and they could not translate that concept into German. They completely struggled over it because it just makes no logical sense. And so when you had to go from something which was kind of misleading in English to go into German, they really struggled back and forth. There were multiple people who had basically failed at translating that idea.
Okay, so mutations break things. Nature can't select. What is happening instead? Because again, we all know that creatures do change and adapt. And so this is a rove beetle right here. And so are all of these here. But what's interesting about the rove beetle is it's eaten by army ants. And so the rove beetle has this interesting ability inside itself. It can change its gene expression. And it can make itself look and behave and even smell like army ants so that it blends in. And it even gets accepted into the nest of these army ants because it mimics them so well. And so it has to have, as you can imagine, all of the genetic information to make these different forms inside itself to start with in order to be able to adapt in this way. And another example, this is the Hawaiian stick spider. And it's kind of confusing, so I'll explain this to you, but there are 3 different kinds of stick spider. Different colors here, as you can see on the left. And what happens is one of those colors will move to an island, and in its offspring it will make all the different colors. And so evolutionists have said, look, this same evolutionary step keeps happening the same way over and over again. But I think what we are discovering instead here is that we have, again, all of that information is contained within the species to start with to allow it to adapt. And I'll let Dr. Brian Thomas explain this one.
That's not a mutation.
It's not a mutation, no. These spiders make offspring that is able to fill, as he'll tell us here in a minute, each of these environments he's going to describe. And so any of those spiders that make it from one island to the next can then produce all the different varieties.
Nature Park on the island of Maui. And why are we here? There's a little tiny spider, the genus Ariadnes, and it's called the Hawaiian stick spider. And it's fascinating because it changes color. it changes color fast. So this rapid color change happens whenever one of the spiders pioneers the next island. So the Hawaiian stick spider lives on these islands in Hawaii, including here on Maui and in this park. And so researchers have been looking and studying these spiders in this park. We're on the north side of Haleakala. What they're finding is these spiders live on the underside of leaves. If it's gold, they live on black rocks or black areas with the black variety, and then white lichen covers tree trunks in different places. So there's a white variety of this spider. Well, what they've discovered is that the spider will pioneer, let's say a black one pioneers, say, Maui, where we are now. From that black parent, it lays eggs, and then out of the eggs hatch gold, white, black. And so you can have on the same island, you can have two black Ariadnes, and they're not very closely related. One pioneered separately from the other. So this rapid and repeatable and targeted adaptation, if you will, or expression of trait change, this is happening really in contrast to the way that Darwin described his theory of trait changes or adaptations. He said that natural selection explains it all, and he even gave a test. He said if trait changes happen fast, then it doesn't fit natural selection. He said natural selection has to happen by slow, gradual, step-by-step processes, and it's not at all what we see when we look at these little spiders. They're happening, these trait changes are happening fast, and they've happened every time that these creatures have pioneered each of the islands in the island.
So they're making these changes so quickly, and then they are potentially, they're reversible changes as well that are going on. They're doing some research right now with blind cavefish and finding that you can breed blind cavefish and make normal fish again in the right environments. And so it's
adaptability rather than a natural selection process. They adapt to their environment. Their environment change, they change to adapt to the environment. It has nothing to do with some lack of thing. How quickly they need to adapt to survive. But they're adapting. They're not taking anyway.
It's like they have the information inside them already to be able to adapt to these changes in their environment. Exactly. And so that is what we're finding in genetics, but we're kind of just starting to emerge from this kind of idea of this mutation, natural selection thing that's been the popular idea for so long now, that we've kind of missed stuff like this.
Because he would say that's their way to continue to survive. And once they're in that environment, if the environment changes, they're going out. They can't. Darwin did not accept the adaptability.
Right.
But,
Right. Exactly. Yeah. So the idea of evolution is that it, not an individual can adapt, but that an entire species has to adapt over very long periods of time. And what this is showing is that that's not true at all. I mean, this single spider can lay eggs that create all of these traits again. And so, again, it's just pointing us back to this idea that all these creatures that we see are already contain the information they need to fill the earth, if you will. All right, so this is kind of a very, very new model, but the Institute for Creation Research out of Dallas, Texas, they're putting forth this idea that rather than shaping organisms, the environment is a passive stimulus, and that the organisms themselves have complex mechanisms for adaptation already inside them, engineered by the Lord Jesus. To me, I think what an incredibly more complete understanding of adaptation to think that God pre-planned the ability of animals to adapt to the environmental changes they would face in order to be able to thrive in variable conditions. You see, he is an ingenious creator. Who has Genesis 1, 20 and 22?
Then God said, "Let the waters swim with fish and other life. Let the skies be filled with birds of every kind." So God created great sea creatures and every living thing that scurries and swarms in the water, and every sort of bird, each producing offspring of the same kind. And God saw that it was good. Then God blessed them, saying, "Be fruitful and multiply. Let the fish fill the seas, and let the birds multiply on the earth."
Yeah, much like our understanding of Isaiah was informed by the understanding of the complexity of genetics, I think an understanding of God's blessing, be fruitful and multiply and fill the earth, is informed by understanding how creatures are given the information inside them that they need to do that. Who has Genesis 1:24?
And God said, "Let the land produce living creatures according to their kinds, the livestock, the creatures that move along the ground, and the wild animals, each according to its kind." And it was so.
And then who has Romans 1:20?
For as invisible attributes, namely his internal power and divine nature, have been clearly perceived ever since the creation of the world, and the things that have been made, so they are without excuse.
Yeah, so we've read that passage a little bit more in depth before today, but I wanted to remind us again that Paul tells us that the attributes of God are perceived in the things that have been made. All right, so what can we learn then about human history from modern genetics? And this I think is exciting. There's an article that's also about this. You can pick up on your way out or scan the code at the end. But so this is fascinating, right? The world population has only doubled 31.5 times from an original pair. It gives us about 6,500 years of time if we calculate it. In fact, some creationists, including Dr. Carter, who we heard from earlier, did an extremely complex computer modeling of the population growth of the Earth. And as they found here, taking into account all these variables, that it was trivial to obtain the current world population of about 7 billion people at the time from a founding group of Noah's 3 sons and their wives. So real quickly, I want to explain to you this idea. The inside the cell is a structure called a mitochondria, and it makes energy, but it is something that you get only from your mother, while everything else is a combination of things from your mom and your dad. Your mitochondria come only from your mom. And mitochondria have their own DNA. And so we can trace these back, and this is a map over here of mitochondrial lineage of the human population currently. And one of the fascinating things is you'll see that the ancestry of our mothers converge on 3 distinct points. And why would that be? Except that we have essentially 3 mothers that got off the ark, right? Noah's sons and wives, who were presumably from outside the family, we'll say. And then if we trace back further and we look for what they call mitochondrial Eve, and that would be to try to find what was the original mitochondrial DNA, we find that our mutation rates needed some recalibrating, and evolutionists became concerned because the original mother of all people would only have been about 6,000 years in the past. And that concerns them because that doesn't fit their story. And similarly, we can track male ancestry through Y chromosomes. These are human chromosomes. If you don't know, you have 23 pairs of chromosomes. And if you're a guy, you've got a Y. And this is something, obviously, you only get from your dad. So similarly, we can track Y chromosome lineages. The rest of our DNA is so mixed up from all of our mixing from our parents it becomes very difficult. But we can track these Y chromosome lineages here of all the people groups on the Earth and kind of track them back in their ancestry and see what we find is that we only have about 4,500 years of human paternal inheritance. In other words, we all had the same dad 4,500 years ago. Anybody guess what his name was? Shout it out, somebody. Probably Noah, right? Doesn't that make sense? So, I think we're going to skip over this one for time. This is Dr. Carter again explaining how this same genetics.
Now I stole this from a government
It works when I don't want it to, and it doesn't work when I do. But we're going to skip over that one for the sake of time, but he's explaining again how the population growth is exactly what we would predict, and that it's ludicrous to think that that the time frame of human existence would only result in the number of people we have today, as big a number as that seems. If you want to hear more about that, it's also similarly described in that article that is available on the table or when you scan the code at the end. So it's truly all in the genes, right? Well, the proof might be in the genes. See, I want to show you a study. First you've got to imagine, okay, what would evolution predict about genetics? Because science, of course, is about making predictions and then doing an experiment to see if the data fits your model. So if evolution were a true model, we might predict, one, that we might have a common ancestor, that if we have a common ancestor, possibly all species might have the same mitochondrial DNA. Two, there might be a continuum of intermediate genes between the species. And three, that species would exhibit genetic evidence of being very old. And that for humans would have evolved from a large population of non-humans. So we would have many ancestors. Now, some have called this the largest study of evolution. It was published in the Journal of Human Evolution, and it involved 5 million genetic specimens from 100,000 different species. And you see, they came to 5 important conclusions. Let's see if they match the predictions. You see here, they find that genetically, most species have arose recently. They find here, and this is what this chart shows, that there's no overlap between species. There's no intermediates between chimps and bonobos and humans, for example. That's what these red squares show. There's no overlap genetically. In fact, they show that, weirdly, all species went through a recent near-extinction event. And then, most concerningly, they demonstrated that it was impossible to rule out humans originating from a single founding pair. So this was published in May of 2018, and by December of 2018, they realized their error. So they added this disclaimer, which I think is hilarious. This study is grounded in and strongly supports Darwinian evolution. And we don't propose a single atom or even we don't propose any catastrophic events. And yet, what did their data show? Except that likely there was a single founding pair, that all species went through a near extinction event in the recent past, that there's no overlap between species, and that there were no intermediates in between them. So, what is the truth here today? The truth is that science is showing us that our genetics exhibit complex creativity beyond our ability to mimic with modern computers. Mutations remove information from genes. natural selection would also remove genetic information from populations if it worked. Creatures have incredible adaptability built in. Human genetics supports a biblical timeline, and the findings of evolutionary genetics fit creationist predictions. Who has Psalm 139?
Were you created by endless being? You knit me together in my mother's womb. I praise you because I am fearfully and wonderfully made. Your works are wonderful, I know that full well. My frame was not hidden from you when I was made in a secret place, when I was woven together in the depths of the earth. Your eyes saw my unformed body. All the days ordained for me were written in your book, before one of them came to me.
You see, the psalmist here tells us that we have a personal creator, that he made all of our delicate inner parts. And so today, I hope that our study of genetics and DNA helps you understand what that might mean. So next week, we're going to be moving beyond creation. If you guys are sick of talking about Genesis, come on back, it'll be different. But our theme for next week will be archaeology. I have to say it is my favorite week of this entire class. So I just think it's so amazing to look at actual sites and artifacts that we see in Scripture. These events happening, and then we go and we look and we dig in the spot and we find those things. And so I hope you will also continue to discover that we can trust the Bible, that it contains the true history of the world. Feel free to scan these codes if you want to check out any of these resources. There's a few more copies of the article out there. Also a schedule for the remaining weeks if you need one of those. But let me pray and we'll get out of here. Father, thank you for the chance to see that your creativity is immense. Thank you for the chance to understand that your word is true. And thank you for the chance now to worship together. In Jesus' name.